Mr. Bondi here with another biology lesson, this time looking at DNA structure. And before we dive into the structure of DNA, let's remind ourselves of what DNA is and what it's used for. DNA is it's a polymer made of many individual monomers, those being nucleotides. We'll get into that in a little bit more detail in a minute. But it's found inside of the nucleus, and its job really is the set of instructions to make proteins. We haven't gone into this yet. We will cover this in the future video, the process of transcription and translation, but it's the two-step process of taking DNA and then turning it into proteins. A gene is just a section of DNA, and it gets turned into mRNA, and then that mRNA gets turned into an actual protein, which will then do all of the things that cells need to get accomplished. Ultimately, again, DNA is like a cookbook, and each gene is a recipe that makes a different dish or a different protein. Now let's take a look at all of the chromosomes that you can see within a human. This is called a karyotype. And in this karyotype here, we're specifically looking at a karyotype of a woman, and it's actually a woman with Down syndrome. If you look down here at chromosome number 21, there's 3 copies of chromosome 21, which is trisomy 21, so there's an extra copy there. And this is a woman because there's 2 X chromosomes and there's no Y chromosome. But the key thing to take note of here is that there's 23 different kinds of chromosomes, and you should have 2 of each. Now, again, one of those chromosomes would have come from your mom, and the other ones would come from your dad. So you would have a full set of 23 chromosomes from your mom and a full set of 23 chromosomes from your dad. Chromosomes are just big, long pieces of DNA. There's lots of genes within them. So if we take a look at more of a zoom in on a piece of a chromosome, we start over here with looking at the cell. Zooming in on the cell's nucleus is where we'll find the DNA. Now this chromosome is a double chromosome. It's been duplicated so that each half of it is identical after S phase. And so each half of this chromosome here are called sister chromatids. But this chromosome was coiled up in the process in prophase. So if we look at a little bit more detail of these coils, we find chromatin, which is that long, thin version of chromosomes, or long, thin version of DNA, I should say. And this is the type of DNA that you'll find when the cell is in interphase, when it's just doing its normal job. That type of DNA is in the usable or readable form. Now, chromatin gets wrapped around tightly around these little protein balls that are called histones, and they just allow the DNA to be more tightly coiled up, so it just takes up less space. We look even further, that's where we get to that classic double helix structure, that twisting rope ladder, as it were. That's really the base structure of DNA. And then finally, we get all the way down to the base of it. We can get down to the nucleotides. And then what we're seeing across the middle here in these colored spheres or ovals are the bases, which really provide the code then for the DNA. So let's remind ourselves of what a nucleotide is and what it looks like. Nucleotides are the building blocks of nucleic acids. In DNA, there's 4 different nucleotides that are used: adenine, thymine, guanine, and cytosine. In RNA, we throw in uracil in replacement of thymine. But the base structure of every nucleotide is the same, and that's they have a 5-carbon sugar. In this case, we're talking about DNA. This 5-carbon sugar right here is the sugar deoxyribose, and then they have a phosphate group. The base over here is interchangeable based upon which type of nucleotide we're talking about. In this image here, we're looking at adenine, and it has its structure here. Now take note, there's a lot of nitrogen inside of the bases, and so oftentimes they'll be referred to as nitrogenous bases or nitrogen-containing bases. The way a nucleotide is assembled is always the same. The sugar is the kind of like the central linchpin, if you will, of the structure, with the phosphate coming off of it and the base coming off of it as well. The base and the phosphate do not connect to each other. Now again, these nucleotides are going to be linked together end to end to form what we call a polymer, or in this case, DNA. So if we take a look at this structure here, specifically looking at this image right here, we can see lots of nucleotides. Remember that a nucleotide is a sugar, a phosphate, and a base. Now in this case, this base is guanine. We can see it down here in our key at the bottom. So a sugar, a phosphate, and a base. This right here is just one nucleotide. So we can see several nucleotides present. Here's another nucleotide and another one, and then the beginnings of another one. The phosphate's kind of cut off from this one. But you can see on this side, there's 1, 2, 3, 4 nucleotides. On this side, there would also be 4 nucleotides. So there's 8 nucleotides present to form this very small piece of a nucleic acid, or DNA. Now, some other things to take note of is the way that this structure is put together. It's like there's two halves. There's a strand of nucleotides on this side and another strand of nucleotides on this side. Now, the way in which they match up is very key. In this case, if we take a look again, here's a guanine. Guanine will always match up with cytosine. Cytosine must be on the opposite side of it. They're called complementary base pairs. Complementary in that they go well together. Now, adenine always pairs up with thymine. And then if we continue this up, we can see all the pairs going up the rest of the way in this chain. But it must always be this order. Guanine must always pair with cytosine. Adenine must always pair with thymine. Now, these bases are held together across the middle with things called hydrogen bonds. Now, hydrogen bonds are a weak attractive force that keeps these two halves of the DNA complex together. Now, eventually, these hydrogen bonds will get broken when the DNA gets used, so it's useful that these are weaker bonds and not stronger bonds. If we take a look at all of the chemical bonds that are illustrated over here on the sides, like the bonds, for example, between the sugar and the phosphate, or the bonds between the sugar and the bases. These chemical bonds are covalent bonds, and they're very strong bonds. Those are the types of chemical bonds where electrons are shared back and forth between atoms. Those are very strong bonds. We don't want those types of bonds down the middle here because it'd be way too difficult to break. This is really useful, again, when the DNA is going to replicate. It's also really useful when the DNA is going to be turned into proteins. Let's take a little bit of a closer look at the structure of the sugars. Again, this is deoxyribose, and it's a 5-carbon sugar. A little bit of chemistry reminder for you that carbon always forms 4 chemical bonds, oxygen will always make 2 chemical bonds, and hydrogen makes 1. Now, the lines that are showed in this image here are indicating a chemical bond. So this line right here between these 2 carbons is indicating a chemical bond. There's another one there, another one there, and another one there. So this carbon right here has 4 bonds off of it. It's assumed there's a bond between this oxygen and this hydrogen here. It's just not shown. So again, when we take a look at these carbons, they each have 4. And within this sugar, the carbons get numbered. So this carbon here gets numbered 1. This is carbon number 2. This is carbon number 3. This is carbon number 4. And then carbon number 5 kind of sticks off of this structure that kind of looks like a pentagon. Almost a bit like a pentagon. The reason why I show you this is because these sugars orientate themselves in a very specific way on the sides of the DNA molecule. So again, here's another look at the structure of DNA, and within it, just so you can see where we're at, here's a phosphate, here's a sugar, and here's a base. So there's 1 nucleotide. So then here's another phosphate, another sugar, another base, there's another nucleotide. So the same type of thing that we saw in the first image. You've got 4 nucleotides on this side and 4 nucleotides on this side. But I want you to take note now that the sugars have been labeled. If you take a look over on the left-hand side, we can see the 5th carbon on this sugar is right here. And it's actually the one that will attach to the phosphate group. And I also want you to notice down here at the bottom of it that the 3rd carbon is the one that's exposed to the next which will bond to the next phosphate group beneath it. And it's the opposite on the other side. On this side, the 3 carbons at the end, and then the 5 carbons down here. So essentially they run in completely opposite directions, or they point in opposite directions. A way in which you can kind of remember this, if you look at this right here, across from the oxygen, it's kind of like a point. Almost think of it like an arrowhead, and it's pointing down. This one points down. This one points down. Over on the other side, this one right here is pointing up. This right here is pointing up. This one's pointing up. Again, remember, it's always a sugar to a phosphate, to a sugar, to a phosphate, to a sugar, to a phosphate on the sides of the DNA molecule, or what we call the backbone of the DNA molecule. Now, it may not mean much to you now, but this orientation of how the sugars point is important when we get into the process of DNA replication. And knowing that one strand points down and the other strand points up. It's something that we call their anti-parallel. They're parallel because they run next to each other, but they run in opposite directions. Think of it also like 2 lanes of traffic. If this was a road, cars on this side can only go this way, and cars on this side of the road can only go this way. The structure of DNA is anti-parallel. Here's some fun facts about DNA that if you were to take the DNA out of just one cell and uncoil it, just from one cell, it'd be about 6 feet long, which is really incredible. Then if you took every piece of DNA from inside of every one of your cells and you stretched it out, it would stretch from here to the sun 600 times. That is absolutely incredible. That's just inside of one person. Another thing I find fascinating about DNA is that it's the same 4 nucleotides used in every single living thing. Whether you're an animal, a plant, a fungus, a bacteria, they all use the same bases, A's, T's, C's, and G's. So ultimately the things that make us different is just the quantity of DNA and then the order in which you find those nucleotides, which then generates the proteins that they make. Some other interesting facts is that the organism that's closest to us from a genetic point of view are chimpanzees, and they're about 98.7% the same when you compare their DNA to ours, which is a tremendous amount of similarities. Mice are about 85% the same, even though they don't look so different. They're mammals just like us and have very, very similar internal structures. And then you're about 41% the same as a banana plant, which sounds surprising, but when you really start to think about it, they're ultimately not that different from us. They're made of cells that have the same types of structures inside of them as we do, right? They have a nucleus and mitochondria and Golgi complexes and a cell membrane. They function in a lot of the same ways. Now, of course, they have lots of differences and they live their lives in very different ways, but it's not surprising, or it shouldn't be too surprising, that their DNA is relatively similar to ours. All of this similarity in DNA will eventually lead us back to what we look at in evolution and the idea of us having a common ancestor. So that's our look at DNA structure. Next, we'll get into the idea of protein synthesis or transcription and translation.